Okay, thank you for joining us for today's webinar on designing a microbial biomarker discovery study. I'm Rachel Seeger, the Marketing Director for PhyloTech, and your host for today's webinar. PhyloTech provides comprehensive microbial analysis services to researchers studying the microbial communities inhabiting healthy and diseased areas of the body. We decided to host this webinar today on practical approaches to microbial biomarker study design because we were receiving many questions from researchers aiming to develop novel diagnostic and prognostic signatures and uncover new targets for medical intervention. These questions were on topics such as selecting the appropriate study framework and determining the appropriate cohort size to appropriately power their study. We thought others might have these same types of questions and wanted to host a forum to begin this discussion. Our speaker today is Dr. Janet Warrington, the co-founder and Senior Vice President of Operations and Development for PhyloTech. Dr. Warrington has more than 20 years of experience successfully advancing life science and diagnostic applications to commercialization. Janet is active in advancing the development of standards and best practices for genomic technologies and is a member of the Clinical and Laboratory Standards Molecular Methods Area Committee and an NIH-NCI Office of Biorepository and Biospecimen Research Steering Committee. Janet earned her Ph.D. in biological chemistry from UC Irvine and was a DOE-sponsored postdoctoral fellow in genetics at Stanford University. Today, Dr. Warrington will describe best practices and important steps for uncovering microbial biomarkers you may one day apply at the bedside. At the end of our talk, we'll open up the line to questions, or you can submit a chat at any time using the controls in the bottom right of your window. You can also raise your hand using those controls, and we'll open up the line to make you available to ask your questions. Okay, with that, I will turn this over to Janet.
Thank you, Rachel. I appreciate the generous introduction. And I'd like to thank all of the people joining us today for this webinar. I'm hoping that you'll find this very useful, and I'm really looking forward to building a relationship with you and discussing your particular study when you're ready to share that with us. So I am going to talk about particularly good study design best practices and really focus on the design of a pilot study. These are actually the most challenging studies to design, and I'll talk about why that is as I go through this overview in the next half hour. Until recently, most people didn't fully realize that communities of microbes in and on the human body affect human health and disease states. Over the past several years, tens of thousands of bacteria have been identified and classified, making possible identifying the members of a bacterial community as well as measuring the changes that occur in those communities during disease. Today, I'm going to talk about things to consider in setting up experiments to identify microbial biomarker signatures, practicing good science, and following best practices. In the current era of translational medicine, personalized medicine, and the widespread implementation of genomic technologies, the growing body of knowledge has increased our understanding that the biology of disease is complex. Being able to integrate clinical information with molecular and microbial information lays a powerful foundation for understanding disease. For many diseases, measuring the bacterial community provides an important component to an integrated view of patient status and treatment choices. Also, microbial population information is being used to identify new drug targets and better understand which patient cohorts are likely to respond to treatment, as well as define the mechanism underlying treatment response. Over the past 5 years, there's been a rapid increase in the number of studies reporting important and exciting discoveries in the realm of microbial bacterial community analysis, as shown in this graph on this slide. Many of these studies have examined microbial population diversity by interrogating the 16S ribosomal RNA gene. Some recent examples include an obesity twin study and a study demonstrating a mechanism by which gut bacteria were interfering with the detox mechanism of acetaminophen. Many of these studies are well designed and executed. The findings are robust and will hold up under the scrutiny of scientific and clinical communities. However, some studies will be underpowered, not address the intended question, or not be well executed. Ultimately, and this is one of the great things about science, good science will prevail. Designing a good scientific study is not that hard if you follow good scientific method. And I'm sure you can all think of studies that have made a big splash in the press, and then they disappeared. And either the validation work didn't pan out, the biomarker panel that was identified early was reported basically prematurely and not robustly tested, and those studies just fade away. And it's an expensive experience for any group to go through, and incredibly disappointing, and you just don't want to be in that position. You really want to have a robust study design so that you'll have a successful outcome and that your biomarker panel can ultimately be used in a clinical application. So in the next half hour, I'm going to walk you through some good attributes of study design, and especially with a focus on pilot studies, because I do believe that these are the most challenging studies to set up and to execute on well. Now, it may seem obvious to many of you, but it's a good exercise for anyone contemplating a study to consider first and foremost, what question do I want to answer? And the list on this slide is just a subset of the types of projects that people focus their biomarker discovery around. Now, this list isn't mutually exclusive, but it is very important that when you're designing your pilot study that you really focus on a specific question. For instance, what type of information can you expect to get from the signature? And are your patient samples, the samples that you're using for the study, appropriate for that question? What will the information be used for? Are you looking for a diagnostic signature, or do you already know that these patients are affected and you're looking for subclassification or some type of patient stratification? It's very important to clearly articulate these things up front. The next slide shows a recent example of a very nice study, nicely designed and executed study. This is a cystic fibrosis study that was recently reported by Michael J. Cox and Susan Lynch. And this study demonstrated that lung function status is characterized by specific bacterial community composition, and in particular, the abundance of known and suspected pathogenic species. It appears that the airway microbiome profile correlates with pulmonary status in CF patients and can reveal key species involved in pathogenic processes in individual patients.